So Emily asked me to tell for the beginning something about the history of electrical stimulation in humans and of the human brain, and I won't start with antique usage of electric fish to treat migraine, which has been done really many 100 years ago, but didn't seem so very effective because we don't do this anymore. But this was one of the first applications of electrical current to the brain to do some treatment of neurological symptoms. Then there were other usages of electrical current of the brain, but also of the peripheral nervous system. By Volta and others who showed that when you apply electrical current to a peripheral nerve, you see some twitching of muscles, you see some twitching of extremities, so some phenomenological effects, which made more clear than before that the neural system, that the nervous system is somehow an electrical organ. And in the 18th century, there were, I show this mainly because of the pictures, these were also treatment applications which didn't make it to the 21st century at the end. So here somebody was trying to treat toothache with electrical current stimulation. One could ask by looking at the patient if the electrical stimulation was a bigger problem than the toothache the patient had. And this is an experimental protocol to make a blind person here seeing by electrical current application. It's not delivered if this was really successful, but again, this was not followed up until the next 40, 50, 100, 200 years. Getting more now closer to what we are doing now with electrical and magnetic stimulation. This is a picture from a guy called Sylvanus Thompson from 1910. And these are coils delivering a magnetic pulse to the head of this guy. And he claimed that this had some effects on the brain, but again, the effects were not very prominent. And this is, I think, in 1980 was a breakthrough for non-invasive electrical brain stimulation in humans, a protocol developed by Merton and Morton. What they did was they delivered a very high-intensity, short-lasting pulse to the motor cortex of humans, and if they stimulate it strong enough, my music on the so everyone's awake now again. They stimulate its motor cortex, and if you apply a high voltage stimulus, intensity about 400 to 700 volts for some milliseconds, you can achieve a supersessional activation of the motor cortex, and you can see muscle twitches here at the hand area if you stimulate it. This is called tDCS and was developed in the 1980s. I don't know if anybody received or conducted TES in this room. Okay, so those who did know probably why this is not the standard technique now. It's very painful because it's electrical stimulation, direct electrical stimulation, which activates also all the pain receptors between the electrode and the brain. So if you conduct this, you're relatively fast conditioned. It's a kind of maximum pain you get. So this is not very practical for routine application. And but based on this, it was known that it's possible by non-invasive stimulation to induce current flow sufficiently strong to activate neurons under the electrodes, and this was the starting point for developing transcranial magnetic stimulation, which also works with relatively short-lasting, high-intensity pulses, magnetic pulses, which have the great advantage that they don't activate all the pain receptors between the coil and the brain. And via this high-intensity stimulation, then you induce electrical current flow in the brain and activate neurons under the coil, and then have similar effects, very similar effects to TES. So it's a kind of development of electrical stimulation, how we do it now in a nutshell. For these treatments I have shown you, it's a kind of super-threshold stimulation, and the most important effects, which were shown especially for the old techniques, were phenomenological effects, where they didn't have the electrophysiology to look for EMG or something like this, so they depended with the old techniques on things they could see. And So you have behavioral effects. You have you see effects of the muscles when you stimulate the motor cortex. And from these techniques, I've shown you tDCS is in a way fundamentally different because it's a subthreshold stimulation. You don't activate neurons at a superthreshold level with DC stimulation like we do it, but you modulate resting membrane potential dependent on stimulation polarity, or to be more exactly, dependent on neuronal orientation relatively to in relation to the electrical field direction. You hyper or depolarize neuronal membranes, and you can see here physiological effects. And this is shown again here. So you have here kind of an action potential. There's a threshold for induction of an action potential, and with one stimulation polarity, you modify the resting membrane potential in a hyperpolarizing direction, which means that you need more incoming activity to induce the action potential in the neuron. With the other polarity, you induce depolarization, which means that you need less activity to induce such a superthreshold depolarization, which is then, however, triggered by spontaneous neuronal activity by efferent activation of a neuron. And what you can see here for practical application is that the current flow direction in relation to neuronal orientation is quite important for getting these effects. Okay, it's getting weaker. If you meet the long axis, you will have a depolarization at a certain point of the neuron at dendritic tree, for example, or so. And you get a hyperpolarization at another area, so current has to flow in and current has to flow out. If you if the current electrical current field is oblique to the neuronal orientation, you will get de and hyperpolarization at a very small area of a neuron, which will result in a zero effect. And so you have to meet the neuron in a certain axis to get effects of the subthreshold depolarization, and this is more important than for superthreshold activation, where if you do a superthreshold depolarization here, you will get an action potential. But if you do it at a subthreshold tonic level, you depend more or less completely on meeting the right axis of the neuron. And also, DC stimulation is not quite new, so we didn't invent this in Göttingen, or Alberto Priori didn't invent this. In Milan, this is a technique which was already used in the 50s and 1960s. Is work of a group in London, a group by Lynn Bindman, and they did DC stimulation not transcranially, but with the electrodes placed on the brain or intracortical electrodes in anesthetized rats. And what you can see here is baseline activity, so spontaneous activity of the sensory-motor cortex of the rat, and here with cathodal stimulation, you see that the activity of the cortex is largely reduced by during tonic stimulation, during tonic cathodal stimulation of this cortex. And here you can see with anodal stimulation as an enhancement of spontaneous activity. And you see this is not induced activity because this is quite chaotic. You can see some bursts here, but it's not regular. So it's not induced activity, it's enhanced spontaneous activity. Okay, that's interesting. But what's more interesting about the technique is that these effects are not only present during stimulation, but you induce after effects of the stimulation. Again, experiments in rat from the same group. And here they didn't stimulate only for a few minutes or few seconds, but for 10 minutes here 2 times. Anodal stimulation, which enhanced spontaneous activity. You can see here baseline activity. With 10 minutes stimulation, you can see that activation is enhanced, at least a bit. Variability is also enhanced. You can see here that with the second stimulation, there's a further enhancement of spontaneous activity, and this did last for many hours after the end of stimulation. Here, more than 6 hours after the end of stimulation, you can see these after effects of stimulation. And for cathodal stimulation, you can see antagonistic effects. Baseline activity here. Again, 10 minutes stimulation, you can see that spontaneous activity is largely reduced for more than 1 hour after stimulation. So if you look at this spontaneous activity, and I could show you the same for excitability, this looks quite similar to what is known now as long-term potentiation and long-term depression. And they were 10 years too early, maybe. So this wasn't a technique which was conducted later in many labs. There were some labs in the 80s and in the 90s, but in animal experiments, these now standard LTP, LTD induction protocols are much more easier to apply. So this supersessional stimulations, and you have standardized protocols, and that's a big with these large electrodes and somehow difficult preparations and also heterogeneous effects due to electrode position. It's a bit more tricky. But essentially, it's relatively similar, and the mechanisms which they explored in animal experiments seem to be also relatively similar to long-term potentiation and long-term depression. So, of course, these after effects were interesting for us because many neuropsychiatric diseases are combined with or are associated with pathological alterations of neuronal excitability, of activity, and of plasticity. So if you can induce long-lasting alterations of neuroplasticity, this might be a point to modulate, to ameliorate symptoms in these patients. There were also studies also in the 60s and in the 70s. Model calculations, we will hear a lot more about models by Maren Dixon in the next talk. But these early models were also not so bad, and they were validated in animal experiments. Here, a study from Russian Driscoll, and validated also in humans. These were pre-surgical epilepsy patients with foramen ovale electrodes in the head, and they showed that under the electrodes about 50% of the transcranially applied direct currents reach the brain, which is more than one would think would get to the brain when you don't have this data and when you just make a spontaneous guess. So this was one point where we got even more excited and then started with the stimulation, as you can see, some years ago. And this is a picture from 1999, so I'm not getting older faster than most of the people. This is a kind of so this was a more or less classical arrangement for motor cortex stimulation we conducted. You have this relatively, they show some features. You see this relatively large electrodes on the head with a return electrode, also relatively large, on contralateral forehead. This is a relatively weak stimulation, so with these were 34 square centimeters electrodes. With these electrode sizes, we applied 1 milliamp of stimulation. This was a custom-built stimulator at this time, and you can see that the stimulation is not painful. So it's this intensity with this electrode size. You feel a light tingling under the electrodes, especially at the beginning of stimulation. But this is, I can tell you, qualitatively different from what you get with tDCS. Okay, so why this electrode position? We were a bit cautious at the beginning, stimulated for 4 seconds, not longer at this time, and applied a TMS stimulus of the motor cortex immediately before the end of this 4-second stimulation. With this TMS stimulus, you get a muscle twitch, and the amplitude of the muscle twitch is an index of excitability of the motor cortex. And we tested different electrode positions here for this short stimulation protocol. So motor cortex contralateral, this is what you're doing normally with tDCS, posterior motor cortex, frontal, occipital motor cortex, and so on. And the only electrode arrangement which resulted in relatively clear effects was motor cortex contralateral supraorbital stimulation, where we got, this is an increase of motor evoked potential amplitudes we got during this short-lasting stimulation with anodal polarity over the motor cortex, and with cathodal stimulation we got a decrease of cortical excitability during this stimulation. So we became a bit more courageous afterwards and stimulated for some minutes then, like they did it in the rat before. And these are the results for anodal stimulation, 5 to 13 minutes stimulation. You can see here again motor evoked potentials versus baseline. So this is an increase of about 50% in this study. You can see that dependent on the stimulation duration, you get longer lasting after effects. a 30-minute stimulation lasting for about an hour or longer after stimulation, but with 5 minutes only for some minutes. So there's a stimulation duration dependent effect. And with cathodal stimulation, where you got during stimulation is excitability diminution, you get antagonistic effects, and excitability reduction of MAP amplitudes. Here was 9 minutes stimulation lasting for about 60 minutes, 7 minutes and 5 minutes stimulation inducing shorter effects. At this time we didn't induce longer lasting effects because so 60 minutes, if you think about 60 minutes excitability alterations, if you work with healthy humans, you don't need more for most of the experimental tasks you're using, so this is absolutely sufficient. And we were a bit anxious at this time. These were the first studies to have, in Göttingen we have many students, but to have many students as subjects and then working around with enhanced or reduced excitability of the motor cortex in the city is something you think about twice maybe. Okay, so these are the effects we get on motor cortex excitability, and then of course the question is, why do we get the effects? And if you think about plasticity, the glutamatergic system is the most important excitatory cortical system in the human brain. GABAergic system, the most important inhibitory system in the brain. So we thought that these transmitters might, glutamate and GABA might be important. Neuromodulators, I don't will tell too much about them, but they have a non-linear important impact on this kind of glutamatergic plasticity might be involved, and because the primary effect is a de- or hyperpolarization, and we thought that voltage-gated ion channels might be involved in the effects of stimulation too. So we looked a bit deeper by pharmacological experiments in the basis of the effects we get. And First, we looked for the effects of for the short effects during stimulation, so for 4-second stimulation, which doesn't induce plasticity. And the rationale of these studies was that when we block voltage-gated ion channels, and the effect we get is de- or hyperpolarization dependent on the stimulation polarity, and we depolarize and block voltage-gated ion channels, then we should abolish the effects of stimulation. If we hyperpolarize, then voltage-gated ion channels shouldn't be active anyhow, and we shouldn't get an effect of the stimulation. And we blocked here sodium channels via carbamazepine, and calcium channels via flunarizine, and you can see that we got the effects we thought we would get. This is placebo medication, and you can see here with anodal stimulation and enhancement of MAP amplitudes. Under carbamazepine, these excitability enhancements are no longer present, and under flunarizine it's diminished, whereas for cathodal stimulation induced excitability diminution we don't have any effect of these drugs on the induced cortical excitability changes. Interestingly, for these acute effects, blocking of the glutamatergic system by dextromethorphan is a drug which maybe some of you know still as a cuff blocker, but essentially it's an NMDA receptor antagonist. You can see there's no effect of dextromethorphan on these excitability changes, and also for lorazepam, a GABAergic agonist, you can see the benzodiazepine, and there's no major effect on the excitability alterations, on the acute excitability alterations induced by tDCS. So the primary effect seems to be membrane polarization dependent. And then we looked for the after effects, and also motor cortex as a model, MAP amplitudes. And what you can see here, we use the same drugs, carbamazepine and flunarizine, to block sodium and calcium channels. And you can see that the after effects of anodal stimulation are abolished via application of carbamazepine. This is placebo medication. This carbamazepine cathodal tDCS effects are not altered by the substance. And with flunarizine, it's more or less the same picture. There's an abolishment of the after effects of the excitability enhancing after effects of tDCS. So you need these primary alterations of membrane potential to get after effects of stimulation. And then for the glutamatergic system, the situation is quite different for the after effects as compared to the effects during stimulation. Well, these are again after effects of anodal stimulation, here 11-minute stimulation. So you can see it's excitability enhancement with cathodal stimulation, the excitability reduction. And you can see that with under dextromethorphan, the excitability alterations induced by DC-stimulation are completely abolished. So this is a strong hint that the glutamatergic system is centrally involved in these after effects of tDCS. So it's a kind of plasticity of glutamatergic neurons. And for lorazepam, so for the GABAergic system, we saw a bit complicated effects. So for cathodal stimulation, there's not a big impact of lorazepam. For anodal stimulation, this is placebo and this is under lorazepam. It seems that the after effects were first abolished and then they were, with a delay, they were somehow enhanced. So, but there was no directional change or no abolishment of the effects, which looked a bit strange at this time, and we had some problems to make sense of this, because normally, if you enhance inhibition, also in animal experiments, glutamatergic plasticity is abolished or reduced. There was a study published some years ago by Charlie Stack from Oxford, who did a MRI spectroscopy study and showed that GABAergic activity is reduced by anodal and by cathodal tDCS. And then somehow it makes sense that you don't get a big overall effect of benzodiazepine of the tDCS-induced excitability changes, because benzodiazepines are enhancing the activity of already active GABAergic neurons or GABAergic receptors, and so if GABAergic activity is reduced, then you shouldn't think of a big effect here. Okay, so having done this, so determinants, what one could say is that we are inducing plasticity of the glutamatergic system, and we need this alterations of membrane potential first to induce this kind of plasticity. I showed you beforehand shortly that neuromodulators have an important impact on tDCS-induced plasticity, and I'm showing you some slides here, not only because we have conducted a lot of studies in this field, but if you think about patients' treatment, for example, or if you think even about experiments in healthy humans, where the concentration of neuromodulators in the brain might interindividually be different, and this is a source of variability, and for patients' treatment it might be quite important to think about it. What's the concentration of neuromodulators, for example, in Parkinson's disease? Dopaminergic system is hyperactive, and if you induce tDCS then in a hyperactive dopaminergic system, you might get similar effects. We have shown here, we applied cyperate, which is a D2 receptor antagonist. You can see here that there's no plasticity induced by tDCS under cyperate in healthy subjects. So there might be different effects in patients as compared to healthy subjects, dependent on the concentrations of neuromodulators in the brain. Interestingly, for L-DOPA, the effect is also not that it's only strengthening after effects of tDCS. There's a strengthening of excitability diminishing after effects of tDCS, but for anodal stimulation, you can see here placebo, you can see a nice excitability enhancement. This is converted into an excitability diminution by 100 milligram L-DOPA in healthy subjects. So don't want to get too much into details, but there's a non-linear concentration-dependent effect of neuromodulators on the after effects of tDCS. It's quite similar for rivastigmine. You can see here that the anodal after effects, this is a choline esterase inhibitor, so it enhances acetylcholine in the brain. You can see anodal effects are reduced. Cathodal effects are a bit prolonged. And this was the first study in acetylcholine. We have done some studies with the nicotinergic with nicotine now, and quite interesting if you're doing experiments in healthy subjects. Nicotine has a prominent impact on plasticity in smokers if they don't smoke for some hours, which you might tell your subjects so don't smoke before the experiments, because this might alter the results of the experiments. We didn't let them smoke for 8 hours, and there was no plasticity in these subjects, so it was completely abolished. which was quite disturbing for me. I'm smoking myself, so I was thinking what this meant. And when they started to smoke again, so we didn't let them smoke, we applied nicotine patches to have a kind of controlled release, and plasticity was reintroduced. So also for the cholinergic system, there's an important contribution, important modulatory effect on stimulation-induced plasticity. And interestingly, for the serotonergic system, you have quite different results. This is cathodal stimulation with placebo medication. So you can see here the only excitability diminution with tDCS, and under citalopram, 20 milligrams, this excitability diminution was converted into a facilitation, and the anodal tDCS-induced facilitation was strengthened in healthy subjects, which in a way, don't know how much you are aware about concepts of origin of depression, and diminished long-term potentiation is an actual concept that this might contribute to this disease. And actually what citalopram seems to do is that it shifts all plasticity you are inducing in the direction of excitability enhancement. So this might be one mechanism why the substance works in depression. Okay, so to conclude, we have shown that the glutamatergic system is involved in the effects of tDCS. GABA seems to have a contribution because it's diminished via tDCS, its concentration is diminished, and you have an impact of neuromodulators. And this is we couldn't explore this cascade so far completely in experiments in humans, but the pattern seems to be similar as in animal experiments, that you increase intracellular calcium. So when we reduce calcium concentration, intracellular calcium concentration, when we block calcium channels, and we don't get plasticity. And depending on the calcium concentration, you can induce an excitability enhancement or an excitability diminution, a neuroplastic excitability enhancement or diminution in the human brain. And this is transmitted via enzyme cascades, via insertion or removal of AMPA receptors from the subsynaptic brain, so subsynaptic membrane. So if you enhance AMPA receptor concentration with the same glutamatergic concentration, you will have larger postsynaptic depolarization. If you reduce the number of AMPA receptors, you will have less depolarization. And this is the most probable mechanism how tDCS works with regard to induction of neuroplasticity. Okay, having said this, then the question is, what are the determinants of the effects of tDCS? And some of our first experiments, we played around a bit with stimulation intensity and stimulation duration. And you can see here, this was 5 minutes stimulation, that stimulation intensity does play a role. So if you stimulate with these are all these 35 square centimeters electrode, with these electrodes between 0.2 and 1 milliamp, you get with stronger stimulation, you get larger effects of tDCS. This was 5 minutes stimulation, so relatively short-lasting effects. But you can see here that there's more or less a linear dependence on stimulation intensity. And you stimulate with 1 milliamp for 1 to 5 minutes, you can see similar increases of the effects induced by enhancing stimulation duration. So stimulation duration and intensity seems to play a role. And the idea why this is the case is derived here from a concept developed by Lissman and colleagues. Again, that there's a calcium concentration dependent induction of long-term depression or long-term potentiation. And with a small enhancement of calcium, you are in the long-term depression range, which a larger enhancement of concentration, you are in the long-term potentiation range. And then here's a no man's land where you induce no plasticity. There might be another no man's land where it's a kind of counter-regulation, and too much calcium concentration will lead to antagonistic mechanisms reducing plasticity. And so it might well be that via prolonging stimulation duration and intensity, you are getting more in the center of this LTP or LTD induction range. But this concept means also that you have at a certain stimulation intensity or duration, you will get an overactivation of the system, and then you might get nonlinear antagonistic effects of the stimulation. And this is one figure from a study we have done, which is published this year, where we stimulated anodally. Here was 13 minutes and here was 26 minutes uninterrupted. And you can see when we stimulate 26 minutes in healthy subjects, you don't get any excitability enhancement anymore with anodal stimulation, you get an excitability diminution. So conversion of the effects, and this might be the case because you're here in no man's land too. Interestingly, so because we thought then this might be too much calcium then, so we blocked calcium channels via flunarizine. So this was under placebo medication, this was with flunarizine, and you can see here that this excitability diminution is abolished. So this seems to be a calcium-dependent effect of the stimulation. So if you stimulate too long, you might get not the effects you want to get. And the same might be the case if you stimulate too strong. This is 2 milliamp stimulation of the motor cortex, anodal and cathodal stimulation. This both is 2 milliamp stimulation. And for 20 minutes. So this is what is normally done in, yes?
Yeah, you said 2 milliamps of cortical stimulation. Do you mean milliamps at the scalp or actually on the brain?
At the scalp. At the scalp. So it's always stimulation intensity of the stimulator. So it gets us 2 milliamp at the electrodes level.
Do you have any idea what an optimal value at the brain would be?
That's tricky, and maybe Marom can say more about this in his talk. But so it's, I think, in principle, so you can model what gets in and where, but there's limited physiological data. So this would be important, but I think also that if you want to prove this terminally, you will need animal experiments or something like this, so to show this directly. So we can just titrate in a way. So 20 minutes, 2 milliamp stimulation, and with both anodal and cathodal stimulation, we got an excitability enhancement here. And we thought then this might be due to that you enhance calcium concentration with cathodal stimulation then to a level where you get into this LTP range. We also thought that maybe we had a strange subject group, so did stimulate the same subjects with 1 milliamp intensity, and then we got this excitability diminution we see normally also in the other studies. So if you stimulate too strong, you might get also different effects of stimulation, maybe induced by the strength of stimulation, but you are also getting deeper with your electrical fields with this intensity, so this might be also a point why you get antagonistic effects. Okay. So how then, what might be an option to induce longer lasting effects? What I've shown you until now, there's a conception of early LTP and late phase LTP in animal experiments. And so the duration of the excitability alterations we had here were all about maximum 1 hour, 120 minutes or something like this after stimulation. So question was for us if you could induce some late phase effects in humans also. In animal experiments, one of the standard protocols is that you do repetitive stimulation, so one induction of plasticity first, then within a time window of about 30 minutes you conduct another plasticity induction procedure, and then you induce those late phase protein synthesis and dependent effects which lead to permanent alterations of excitability. So what we did here was these were from the design very similar experiments. We applied stimulation durations which induce long-lasting after effects about an hour of duration in healthy subjects, so 9 minutes stimulation with cathodal, 30 minutes stimulation with anodal stimulation. Then simply doubled stimulation duration in the first experiment. Then did repeated stimulation with these 9 and 30 minutes stimulation with 3 and 20 minutes intervals, so within the time window which induces late phase plasticity in animals, and then 3 hours and 24 hours interval. We did these intervals also because these are the intervals which are in most cases applied in clinical studies, so you stimulate your patients 1 time a day for consecutive days. And what you can see here for cathodal stimulation is that simply doubling stimulation duration makes the effects a bit more stable but not much longer. Same is the case, so it's much more stable here, so it's somehow more efficient if you do a second stimulation. It's 3 and 20 minutes, but again it's not lasting over the day you're stimulating. And if you're stimulating with 3 hours or 24 hours interval, you get reduced after effects of the stimulation. So there is some after effects when you do it once a day, but it's not as stable as a single stimulation compared to a single stimulation. So for cathodal stimulation, we didn't get this late phase effects. For anodal stimulation was a bit different. I showed you already this excitability diminution with 26 minutes stimulation. It's a short interval. We get first a bit less stable effects, but then we get effects stable until the evening after the stimulation day, so more than 24 hours stable, and it's not going back here. And for the long interval, you again get somehow weaker effects. So again, if you play around with repetition intervals, you can relevantly, at least in our healthy subjects, in the motor cortex, relevantly prolong the duration of plasticity induced by tDCS. Okay, so to conclude here, tDCS is well suited to model non-focal plasticity in the human brain. So we have this large electrode, so it's not very focal. And we get non-linear effects dependent on stimulation duration and strength. So we have to take care when we introduce stimulation protocols that it's not only if we have weak effects, stimulate longer, stimulate stronger, and you will get better effects. There might be some non-linear stuff. Okay, question then, do we have remote effects of tDCS? What I've shown here until now were regional effects more or less under the electrodes. And it's this relatively early study conducted by Nicholas Lang, the time he was in London, a PET study with tDCS. You can see here with anodal stimulation, with cathodal stimulation. Don't want to get too much into details, but you can see effects on the activity of a range of cortical and subcortical regions, including also the brainstem, many cortical regions, which are in a way polarity dependent but not absolutely homogeneous. And of course, this could be caused by connectivity, but this could be also caused by spread of electrical current flow. And this wasn't clear at this time. This was the first study where we looked for remote effects of tDCS. It was again tDCS combined with TMS. Is stimulated with relatively small electrodes, so 3.5 square centimeters, premotor cortex and M1. And when we stimulated M1, we got an excitability enhancement with this single pulse TMS elicited MEP amplitudes. When we stimulated the premotor cortex, it didn't get this. And what we knew from previous experiments was that we don't get only this cortico-spinal effects with single pulse TMS, but with double pulse TMS stimulation, you get also effects of the stimulation on intracortical excitability, so intracortical inhibition and facilitation. With the large electrodes over the primary motor cortex, we get both effects with motor cortex stimulation. With the small electrodes, we didn't get here these intracortical effects. Don't want to get into details, but it looks quite similar, independent from what we are doing. Anodal cathodal this was anodal stimulation versus baseline at different time points. So this was quite similar. Interestingly, when we did premotor stimulation, this is intracortical inhibition, and here is intracortical facilitation. This is baseline. You can see that there is, like we had with the large electrode in the primary motor cortex, a diminution of intracortical inhibition and enhancement of intracortical facilitation. So what we get with the large electrode is motor stimulation. With the small electrode, we get one parameter altered with premotor stimulation and another one with primary motor cortex stimulation. So it looks like there's a combined effect of these large electrodes on different neuronal populations, and this might be a hint that with tDCS you don't only stimulate the principal neurons of the primary motor cortex, but you need also stimulation of efferents of motor cortex principal neurons to induce the plasticity effects, which makes sense when you think again about the mechanisms I've told you. And this is a new study where we did parietal stimulation. Parietal cortex is also closely connected to the primary motor cortex. And preliminary results, we stimulated over P3, looked for motor cortex excitability. And here you can see that anodal stimulation enhances cortical excitability, cathodal stimulation reduces it. Sham stimulation does nothing. So relatively similar to the primary motor cortex, but the inhibition seems to be not so strong. And this could this is not quite clear. Could again be current spread, of course, could be connectivity-derived results. We have new results where we just move the electrodes a few centimeters behind and lateral, so not very much, and we didn't get any effects of this prior to stimulation of motor cortex excitability, which might be a bit more a hint for connectivity-driven results. We do now dual coil experiments to probe this further, but so it might be that you're really getting stimulation of one area, relatively prominent effects of another area, which might be connectivity-driven or current spread-driven, and we will explore this further. The other question is, are there really network effects of stimulation? And Raphael Polarity did quite a lot of studies with regards to this MRI studies and also EEG studies. I will show you one example. This is resting MRI. He did first 5 minutes resting state MRI, then 10 minutes anodal stimulation, in this case of the primary motor cortex, contralaterally the cathode over the frontal orbital region, then again 5 minutes resting state MRI, 10 minutes stimulation here, 10 minutes compares this with 10 minutes sham stimulation. Did these calculations, and I will show you here the seed-based approach. He did also graph theoretical things. You can see here, if you put the seed in the left motor cortex where the stimulation electrode was positioned, you can see enhanced connectivity with the premotor cortex and with the parietal cortex. So you can see network effects at the cortical level, and for the right electrode frontal orbital, you can see right insular stimulation. So also here you can see some kind of network effects of stimulation. Did some other studies looking for cortical, subcortical effects also, and for intrinsic motor cortex connectivity, where he could show also network effects. And you can see the same or similar things with EEG. Conceptually quite a similar study. He did resting state EEG, motor task EEG, then 10 minutes tDCS, anodal tDCS or sham tDCS, then again resting state and motor task EEG, did these calculations and came up with this connectivity map. As the electrodes he has done, and you can see that there are alterations in many frequency bands. The most important one seems to be in the gamma frequency range here, high gamma frequency range. And what is shown here is, so this was the electrode position. This is tDCS only, so before, after tDCS. So you don't see on this oscillatory connectivity, you don't see big effects of tDCS, so there's virtually nothing. If you look at the task-dependent effects, this is task performance before and after stimulation, you see a relatively huge increase of motor cortex connected areas connectivity. But in this picture, it's repetitive task performance, so before tDCS and afterwards. So it's a mixture between task-dependent effects and stimulation-dependent effects. This is sham stimulation compared to real anodal tDCS after the stimulation, so second task performance. You can see here what the tDCS contributes to this enhanced network connectivity, and you can see here that there's quite a lot of motor, premotor and also prefrontal increasement of connectivity by tDCS and this task-dependent oscillatory activity. So there seems to be a synergistic effect of DC stimulation with task performance on network activity. So what I've shown here is that you don't have only regional effects of the stimulation, but you have also network effects of the stimulation, which might be also functionally relevant if you think about this task-dependent effects. Okay, so I've not too much time now. I want to say some words about optimizing effects of tDCS, increasing focality of tDCS and prolonging the duration of after effects. Just also putting together stuff I've taught in the last minutes. So mood is good here in the first studies, but the electrodes are relatively large, and this is of course is not a big problem if you do some basic physiological studies. We only want to look for excitability or pharmacological experiments. Might work nicely, but if you think about you want to study tDCS DCS effects on cognitive tasks, so you need some more focal effects. Question is how to achieve this. One option might be to simply to reduce the size of the electrodes. I've stated this before with premotor stimulation already a bit. It's again motor cortex results. What we did here is that we have here the big electrodes, which covers the ADM and the FDA muscles, from which we recorded motor evoked potentials here during stimulation, also afterwards. And then we reduce the electrode size, only covering the ADM and not anymore the FDI. And the idea was, if we have more focal effects with the smaller electrodes than with the small electrode, we should induce excitability alterations of the muscle representation covered by the big and the small electrodes, but not of the muscle not covered by the small electrodes in the small electrode condition. And this is what at the end we got for the effects during stimulation. Afterwards, this is ADM covered by the big and small electrodes. You can see polarity-dependent excitability alterations with both electrode sizes. For the FDI, it's only with the large electrodes, not for the small electrodes. This is during stimulation, and for the after effects, quite similar. Big and small electrodes alter ADM excitability, but only the big electrodes alter FDI excitability. So reducing the size of the stimulation electrodes might make sense to get more focal effects. This is a bit older modeling study of Miranda and colleagues who show more or less the same, that with a smaller electrode you get more focal effects of stimulation. It's a simplified model, that you might have to stimulate a bit more intense to get at the predefined depth of this area the same effect as with a large electrode. So it's not quite linear, but it shows in principle by the model that that's not quite wrong, that with a smaller electrode you get more focal effects. And next question is, what do you do with a return electrode? In the classic electrode arrangement, this is active, and you don't want this active electrode in many cases. Sometimes it doesn't matter much, but in many cases, if you think about cognitive research again, you don't want this. And we thought, okay, if the effects depend on current density, so more current density means more efficiency of stimulation, let's increase the size of the electrode here, 10 per 10 centimeters, and then we reduce current density under the electrode and might reduce therefore also efficiency of stimulation under this electrode. Did this here, and you can see independent for this motor cortex excitability effects independent from the size of the return electrode. You get identical effects on excitability, and this is a task where we know that the frontopolar cortex is involved in performance, and with the conventional electrode size, so you get improvement of the task, the probabilistic classification task. Increased values show improved performance. So this is normal electrode size where they get better during task performance. It looks a bit noisy, but it's at 2 time points is significant. And with sham stimulation and with the large electrodes there, you don't get any effect. So this is the same as sham and the large electrode. So you make this is big electrodes. This is functionally inefficient in this case. So enlarging the return electrode might make sense to improve focality of stimulation. Another option might be to have an extracephalic return electrode. A study conducted by Vera Moliace some time ago, where the electrode was where it was compared return electrode position at contralateral forehand, upper arm, and you can see that here the excitability alterations were not as big with the upper arm return electrode. This took place also for, here you can see the effects over time. So lower effects with the upper arm electrode, and then she increased intensity to 2 milliamperes there, and what you can see here that these excitability alterations were enhanced by this electrode. So it's possible to have this remote electrode, but you might have to stimulate with larger intensity to get the same effects. And another potentially problematic aspect might be that you have different current flow directions, and you might activate different neuronal populations because of this. This is something nobody really knows, so there's not much physiology. Another option might be, so you have again here this impressive size of the electrodes. A study conducted in Maron Bixen's lab. This modeling you can see, and you can see here this HD tDCS arrangement. Maron will tell much more about this, and you can see here with the model you get much more focal effects with these 5 electrode arrangement. And we've conducted a study where we looked for the efficiency of this new electrode arrangement to induce excitability alterations. You can see here that we did stimulate there with a large and with a small electrode with 2 milliamp. Quite similar effects, maybe a bit higher and a bit longer lasting with a smaller electrode. One has to say, of course, that it's not quite clear if that what you get at a cortical level is identical with 2 milliamp with a large and with a small electrode size. So it might be that this is also due to enhanced efficiency to get current with the same intensity in the brain there. And so it's for me it's unclear if it's more efficient per se or not, but at least it seems to work nicely. So this is, and the models show that it should be much more focal than the stimulation with a large electrode, which is something which has to be tested also physiologically now. Okay, so there are lots of options to enhance the focality of stimulation, and just I don't know if I have 5 minutes, I can tell.
10 minutes.
10 minutes, okay. So we have time for questions. I can tell something about the prolongation of after effects, which might be important, especially if you think about clinical application of tDCS. So what I've shown already, increasing current density and stimulation duration might work at least in a certain range of stimulation intensity and stimulation duration, but not in all ranges. And don't have to get into detail here. So 2 milliamp might convert cathodal after effects, and so repetition might be a point. So this is the study I've shown before. Just experimental details. We did baselines and we did repeated cathodal TDCS, and the other study anodal TDCS. Simply doubled stimulation duration. Then this seemingly important time window is in 30 minutes repetition, and then hours between the repetitions, and you seem to get more stable and or stronger effects when you are operating in this time window of 30 minutes into repetitive stimulation. And what seems to be also the case, I show you here individual results, and it looks that for the short time window, which is here, the effects seem to be more, and especially for the 20 minutes, much less variable as compared to a single stimulation and as compared to these long interstimulation intervals. So in this case, for cathodal stimulation, you don't get excitability alterations lasting for days, but you get more stable, less variable effects of the stimulation. And then for anodal stimulation, you get within this time window, you get these long-lasting effects of stimulation if you do it twice a day. By the way, nothing is known if you do this on consecutive days, of course, so 2 times a day for 10 consecutive days or something like this, which one might think about doing this for clinical studies. And interestingly, for these long-lasting after effects, they seem to be also at least partially dependent on NMDA receptors, so it seems to be a glutamatergic effect, because when we apply dextromethorphan at the day following stimulation, this is without dextromethorphan, this is with dextromethorphan, and you can see that the effects are abolished, but at least partially are recovering the evening, let's say after stimulation day. So it's an effect dependent on the glutamatergic system. And then an option to prolong after effects might be pharmacological intervention. And you can, I just put this together here. D-cycloserine is an NMDA receptor agonist, and you can see here that anodal stimulation after effects are relevantly prolonged for until the evening of the stimulation day, so which next morning, which is about 16-18 hours after the stimulation. D-cycloserine, however, has a problem that it's a partial NMDA receptor agonist, so it works agonistically in a relatively narrow window of concentration, and if you do too much, then it has antagonistic effects. But at least here you get a prolongation, you get a similar prolongation of anodal after effects as amphetamine, which doesn't surprise maybe. And with citalopram, I've already shown enhancement of anodal after effects and also conversion of cathodal stimulation effects to an excitability enhancement. So if you want complete excitability enhancement, citalopram might be a good thing. And there has been conducted a study in depression combining tDCS with serotonin reuptake inhibitor, which had strong effects on stimulation or the drug alone, which is in advance also for this hypothesis. If you want to induce prolonged excitability diminutions, then the dopaminergic system might be a good idea, also dosage dependent. Here's a study with pergolide, where we had after effects until the evening after stimulation with cathodal stimulation compared to about 30 minutes after the stimulation with placebo medication. The same is 100 milligram L-dopa, where we had after effects lasting for about 24 hours after the end of stimulation. So adding pharmacology to stimulation might be a good idea if you want to have longer lasting effects. Okay, so to conclude, you can achieve prolonged effects of stimulation under certain conditions by increasing current density, stimulation duration, repeated stimulation, and also by pharmacological intervention. And that's the end. Many thanks for your attention. And we are doing this all to improve the quality of our football team. It was promoted to Bundesliga last year, so it worked quite well. So we are doing something different also sometimes than brain stimulation. Rahel Polarity did lots of the network studies. Lisa Stixrud just came back from Canada and doing now some things with brain stimulation, Parkinson's disease, and impact on task performance. Min van Koo is a postdoc, one postdoc in my lab, who does the day-to-day PhD supervision, also did lots of the pharmacological stuff. Georgi Batsikate does now nicotine and serotonin, and we don't do beer studies. Many thanks for your attention. Okay, that's it.